Hopefully it's loaded. Okay, I've got a long title too, but I won't use it. I'll just go ahead and plunge right into what I'm going to tell you about today. I wanted to thank Tom and Tony and the other organizers for the invitation. I'm very happy to have a chance to tell you about some of the work we've been doing. I'll be talking about conjugated polymers, as Paul did, but the wrinkle in the cases that I'll be talking about is that these are conjugated polyelectrolytes. On each polymer repeat unit, we have a charge group, either negative or positive, and that what renders these molecules water soluble, and so they have their own set of characters. But what you'll see is that there are some real similarities between what we found with these polyelectrolytes and what Paul and others have found with neutral conjugated polymers. Anyway, I should start with acknowledgments. Interestingly, this work was really started at Los Alamos about 8 years ago when I was working with Duncan McBranch, Mia Hai Chen, and Xingling Wang. And so the work really started there. This work actually led to Duncan and I co-founding QTL Biosystems. A little bit of the work that I'll talk about this afternoon was done at QTL. My co-workers there were Liangdi Liu and Commander Achuthan. And then much of the work I'll talk about is recent work that's been done at UNM with a couple of students working with me, a postdoc, and then colleagues at UNM in the Department of Chemical and Nuclear Engineering, and then colleagues at the University of Florida, Katsu Ogawa and Kirk Shantz, former student of mine. This work has been funded by DARPA and NSF. Okay, so here's what I'm going to tell you about today. I'm going to talk a little bit about super quenching of conjugated polyelectrolytes, and then I'll spend most of the time talking about these conjugated polyelectrolytes on nanoparticles and microspheres. We'll talk about fluorescence quenching and its application. I'll tell you about some recent sensing that we've developed, what we call frustrated super quenching, and then I'll end up talking about these conjugated polyelectrolytes and their biocidal activity, and believe it or not, that probably involves energy transfer as well. Okay, so the first conjugated polyelectrolyte we worked with was this MPS PPV, and I understand there's still people at Los Alamos working with this. And it's polyelectrolyte, it's water soluble, it's a single conjugated molecule, it's conformationally mobile, and what really appealed to us when we started having an opportunity to work with this is it's about the size of a micelle and has about the charge density of a micelle. And we thought it might interact with counterions. It might associate with them, and that might affect them. And so it does. This has a fluorescence efficiency very low, about less than 1% we think in most cases. And of course, it does, like a micelle, readily bind organic and inorganic counterions. So we decided to look at the fluorescence quenching possibilities with this molecule. And the first quencher that we chose was methylviolgen, this organic dication. Now, we had observed earlier that if you take a small organic molecule like transtobine, it forms a ground state complex with methylviolgen, and the fluorescence is quenched. The quenching constant is very modest. It's around 15 for Stern-Boehmer quenching constant. And this is due to the weak association, where every stilbene that's associated with a methylviolgen has its fluorescence quenched. It's just a weak complex. Now we found that we could enhance this in a micelle if we add, for example, an anionic detergent. What that does is concentrate the stilbene and the methylviolgen on the surface of the micelle, and the quenching constant goes up to about 1,000. So we compared that to the MPS PPV, and what we found here is that the quenching constant's up to 10 to the 7th. This is, I think, about the highest quenching constant that had been observed when we published this back in 1999. So we had really dramatic quenching enhancement with the conjugated polymer and this electron transfer quencher. And we decided to see if we could couple this to biosensing opportunity, and what we did was we found that we could do that. But let's talk a little bit about super quenching. What does it do to? Well, first of all, we have the strong binding of a potential quencher counterion to the charged polymer. And we believe again that there's efficient, or relatively efficient energy migration or excitonic delocalization in the polymer. And I'd have to say from our work we have very similar results to what Paul talked about, that in general the excitation seems to be delocalized over something close to 100 to 500 polymer repeat units. You can affect this quite a bit by putting something in the aqueous solution with the polymer. For example, if you add an oppositely charged surfactant at very, very small amounts of surfactant, you find the fluorescence efficiency goes way up. But of course, when it's extended like this in a polymer brush, then we find that there's more difficulty in quenching. The quenching rate constant goes down. But anyway, super quenching, we think, goes somewhat similar to artificial or natural photosynthesis. You have this efficient energy migration, and then you harvest the energy at a trap site where an electron or energy transfer quencher is bound. And we've done a lot of studies with both electron transfer and energy transfer quenchers.
Can I ask you, is this similar or the same or different from what I've heard called amplified fluorescent polymer?
Yeah, whatever. Sure.
No, I'm not that familiar with it. Is it the same mechanism?
Yeah.
Okay.
Okay. So we have a quenching constant of, let's say, 10 to the 7th or 10 to the 8th. I've actually seen them up as high as 10 to the 9th. About, well, we've actually tried to separate out, what do we attribute that high rate constant for quenching, or Sturm-Homer constant. And for example, about 10 to the 7th is due to the strong association. And then we would say somewhere around 10 to the 2nd, or sometime 10 to the 2nd is due to the delocalization. So we also found that, and I'll be talking mostly about supported polymers. One of the problems that we found with the simple polymer in solution is it's subject to interacting with almost anything that you put in there. If you want to do biosensing, you have certainly some problems with non-specific association. On the other hand, the polymers that we're working with now and that we've used to make commercial sensors are mostly polyphenylene ethynylene polymers. They still have charge on each unit. These have much higher fluorescence efficiencies, in part because these polymers tend to be rod-like and don't have so many conformations as the PPB polymers. They have fluorescence efficiencies in the neighborhood of 30 to 50%. And ironically, when we put these down onto supports, we absorb onto a microsphere, we find there's very little quenching of the fluorescence. The fluorescence envelope does change, but there's very little quenching of the fluorescence, and the polymer on the microsphere support is easily quenched by the same kinds of quenchers, oppositely charged electron acceptors or energy acceptors. We also find that while there isn't much quenching for monomers or even small oligomers, when we put these monomers or small oligomers down on a surface so that they associate with each other, we get about the same level of quenching that we get with the polymer. So again, what we found is one quencher can harvest excitation delivered to an ensemble of 500 or more polymer repeat units. So we have developed quite a bit of biosensing, and this was mostly done at QTO. We essentially couple a quench or an unquench of the fluorescence with a biorecognition event, and we use microspheres or nanoparticles as a support. It's usually absorptive coating, and the basis, this has formed the basis of effective sensors for nucleic acid hybridization, for protein sensing, and most importantly for enzyme activity, where there's important drug discovery opportunities. But I won't say anything about that today. I'd like to go on and talk about some recent work that we've been doing. And this involves actually growing polymers, these polyphenylene ethynylene polymers, directly onto glass or silica particles. Essentially what we do is we react the silica particles with put an iodobenzene unit, and then we add the monomers that will give us a polymer. And here's the polymer we normally form. We have actually one of the monomers is a diiodobenzene derivative, and the other is a diethynyl benzene derivatives. So these give you this polymer where you have alternating polyphenylene methionine units. So this is, for example, an anionic polymer that we made, and we've looked at this polymer itself as a sensor, and we wanted to compare how these polymers actually grafted onto beads would behave. What we found has been quite interesting as far as energy and electron transfer. We found that these polymers, as I'll discuss a little bit later, are not, when they're on the beads of this sort, they're not ideal sensors, but you can pursue some sensing applications with them. But we found, we think, more interesting things to do. Okay, if you look at the glass beads we start with, and these are, I guess, 300 nanometer beads, and you see they're very smooth. And when we coat them by growing the polymer on them, you see the surface becomes much rougher. It's very easy to see this with the transmission electron microscopy. You can see they even tend to stick together. This is just an SEM image of the larger particles, and you can again see it's a very ugly looking surface. Interestingly, if we look at the same kind of beads, but with the adsorbed polymer, it looks much more smooth. So there's a really big difference between these polymers that are grown on the surface and the polymers that are adsorbed as a coating on the surface. By confocal microscopy, you can see the fluorescence of the polymer very clearly. You can again see that these beads tend to stick together to some extent. So we can make these surface grafted conjugated polyelectrolytes. You can use various size polymers, various size particles, and you can vary the loading of the polymer. Now, in the one that we've worked with, and I'll show you results with in just a second, we've estimated that by thermogravimetric analysis that you have about 16% of the material of one of these beads is polymer. And we've been able to assert that this is probably around 10 polymer repeat units. It's about 12 nanometers thick. So the polymer fluorescence and excitation are very different from the soluble polymer with the same number of polymer repeat units. The fluorescence excitation spectrum is blue shifted. The fluorescence of the surface grafted conjugated polyelectrolytes is actually shifted to the red. It's very broad and featureless. And the quantum efficiency is probably about half what that is for the parent polymer in solution. It's still maybe 20% or so, but it's definitely reduced. We found that the fluorescence can be quenched by both electron transfer quenchers and also by a series of cationic cyanine dyes, which are good energy transfer acceptors for the polymer. But the interesting thing we find is that if we try to quench the fluorescence with, say, methyl biologen, an organic dication that quenches by charge transfer, what we find is the quenching saturates. There's about 40% of the fluorescence that can't be quenched at all by this. If we compare that with the cyanine dyes, which are about the same size and relatively similar shape to methyl biologen, we find that the fluorescence quenching for several of these monocationic cyanine dyes, which quench by energy transfer, the fluorescence can be completely quenched. So we are imagining now that there must be many sites where there is where you can't get a quencher close enough to quench. So what we suspect is going on is, as indicated here, if we have a charge transfer quenching, for example, using methyl biologen, the quencher cannot get very far into this brush of polymer on the surface because there's just too much steric crowding of the polymer. And so excitons are generated that don't migrate very near a methyl biologen or, for example, even an inorganic ion. And so there's a significant fraction that can be quenched that are near the surface, but there's a significant fraction that can't be quenched where the exciton is buried and the biologen can't get close enough. In contrast, with a 12 nanometer layer, any place you put a energy transfer quencher, you're going to have a good probability of quenching the excitons. So we think that's why the energy transfer quenching is much more effective in this case. Now I mentioned that these polymers are not really good for sensing, at least in the applications that we pursued. I'm going to tell you about some sensing that we have done that involves adsorb coating, where we can actually use this polymer bead as a scaffold to build a bilayer. We try to build a bilayer onto these surface grafted polymers, and we find that we cannot get complete coverage by a lipid under conditions where the adsorb polymer is covered quite well. So what I'm going to tell you now is what we call frustrated superquenching. This is an enzyme activity assay, and we use this adsorbed polymer. This is a we can use this, or we can use a single cationic group, a trimethylammonium group that's a little farther away from the polymer. But this forms a very nice adsorbed layer onto various microsphere supports, and we actually now look at quenching of it with this anionic electron acceptor, anthraquinone disulfonate. And what we find here is when we have this polymer on a microsphere support, we can quench effectively all of the polymer by adding just a small amount of the anthraquinone disulfonate. So the anthraquinone disulfonate associates with the polymer on the microsphere, and the fluorescence is very strongly quenched. Just to show you the polymer, when it's absorbed on a microsphere, it absorbs very broadly with a peak near 430 nanometers and emits at around 500, a little to the red of 500. The surface grafted polymers emit like this, around 560 to almost 600, and they are more to the blue in terms of fluorescence activation. Okay, so what we were interested in doing was to develop an assay for phospholipase, various phospholipases, including phospholipase A2. Phospholipases actually take a phospholipid and remove one or the other carboxylic acid group, and that renders the lipid a relatively poor surfactant and often destroys a bilayer. So phospholipase A2 is water soluble. It's a 120 amino acid peptide, and phospholipase is found, interestingly, in mammalian pancreas and snake and bee venoms. So it has good and bad aspects for humans. So what we expected and observed is that we have the beads coated with the fluorescent polymer, and we add anthraquinone disulfonate. The fluorescence is quenched as the anthraquinone associates with the beads. What we hoped was that if we built a bilayer over this particle-supported polymer and now added anthraquinone disulfonate, that there wouldn't be very effective fluorescence quenching. But now, if we added phospholipase and the phospholipase removed the bilayer, then we'd see quenching. And that's essentially what we found. We've actually been doing this by flow cytometry and also by conventional fluorescence. The flow cytometry is nice for this. It's very easy to do. And the interesting thing, the only way this will work is in the flow cytometry, if you see quenching, that means the quencher is associated with the particle surface and is not swept away readily by the flow process. In fact, what we find is we get very similar results monitoring fluorescence quenching by flow cytometry and also by conventional fluorescence. So here's the fluorescence of the polymer bead, and here's the fluorescence after we add a small amount of anthraquinone disulfonate. Here's the fluorescence again, strong fluorescence, where we have an overcoat of dimeristoyl phosphatidylglycerol. And when we add now anthraquinone sulfonate, we find in most cases that we don't see total lack of quenching. There is some quenching, which probably indicates that the bilayer isn't perfectly surrounding the polymer. And there's a small amount of enhancement of fluorescence, but there isn't very much quenching. And this small amount of quenching that we get is saturated very quickly. Doesn't go any further with time or anything of that sort. Now, if we take the beads that have polymer and DMPG on it, and then we add the phospholipase, and then add anthraquinone, you can see that we have comparable quenching to what we have for the polymer itself. So clearly, this is a good assay for the activity of the phospholipase. In fact, what we can do is run a EC50 course for this reaction, and what we get is an EC50 of about 2.5 nanomolar for the phospholipase, which compares very well for what we find in the literature, right around 2 nanomolar. What we've been doing recently is a similar experiment, and this is we coat the polymer-coated microspheres with a bilayer. This time, what we use is a small amount of the bilayer with the rhodamine fluorophore in there. And what we find is, when we do that, the polymer fluorescence is almost completely quenched. And then, of course, as we add phospholipase and we remove the bilayer, we can see the polymer fluorescence comes up. We're just doing these experiments right now. This just shows that when you add Triton in the presence of the anthraquinone in the presence of this, and remove the bilayer, then the fluorescence goes down. Okay, the last thing I'd like to tell you about today is our work with the polymer as a biocide. And we were initially interested in this polymer. This is a cationic polymer with quaternary ammonium groups. And we knew that a lot of quaternary ammonium surfactants are effective as biocides, and we thought maybe this would be the case. And we also thought, since the polymer absorbs light in the visible, we might have a visible light enhanced biocidal activity. Well, in fact, what we found is that, adding the polymer to suspensions of various bacteria and exposure to visible light did result in killing. We could also take the polymer on a microsphere support and expose it to suspension of bacteria, and we found there was some killing under those conditions. So the question is, why and how much? Anyway, what we found is very, very clear that if we just take the polymer in solution and add a suspension of bacteria with it, just mix it, shake, and so forth, the polymer coats the bacteria and maybe even ingested by them. For example, here's a sample of E. coli that's been exposed to the polymer by phase contrast microscopy, and then here's the same suspension, the same area looked at by fluorescence microscopy. You can clearly see that the fluorescent spots associate with the bacteria, and the bacteria is now fluorescent because of its coat of the polymer. We can take, for example, anthrax spores and look at the coating of the polymer on the spores just by having excess polymer present. And what we get is slightly more than monolayer coverage of the spores, and the spores are, again, very highly fluorescent. They're also dead as soon as they're exposed to visible light. So here's an experiment where we add spores to the solution of the polymer, monitor the decrease in absorbance due to the coated spores, remove the coated spores and suspend them in water and expose them to ambient light, and nearly 100% of the spores are killed with just minutes of ambient irradiation. So we've looked at this in a qualitative fashion, and we think that the origin of biocidal activity may be due to singlet oxygen generation at the surface of bacteria. We know that generating singlet oxygen from the polymer by the beads or polymer itself would not result in any killing of bacteria due to the fact that the lifetime of singlet oxygen and the rate it could travel are not sufficient to deactivate or to counter the bacterium and kill it. We find if we coat, for example, microspheres with known singlet oxygen sensitizers, we get very similar biocidal activity. We think that it's an interfacial singlet oxygen generation, and there's been a lot of speculation in the literature, and actually some fairly good cases made by workers, for example, at Scripps in San Diego, that you generate singlet oxygen in an interfacial environment where you have water, but not very much of it, and not bulk water, is that you can get nucleophilic attack of the water on the single oxygen and formation of more corrosive reactive oxygen species. On the other hand, we can't rule out the fact at this point that the light-induced biocidal activity couldn't occur by a charge transfer mechanism. We have, of course, the polymer that's cationic, and we have halide ions, and so a reasonable possibility would be an electron transfer from, say, a chloride ion to the excited polymer, and then escape of the chlorine atom, and that would be very reactive towards bacteria. We're trying to do experiments now, but it's not as simple as we'd like, because of course we need to find a bacterium that's killed by the polymer, but also is not killed by the absence of oxygen. And we actually have worked with one bacterium that we think will fit the bill. I'm coming back, though, now at the end to the surface-grafted conjugated polymers, and where we find they may be very useful is, in fact, when we compare when we make the cationic counterparts of the conjugated polymers I showed you earlier, where we have the dependent quaternary ammonium groups, and so we have the cationic polymer grown off. And we find, once again, that if we look at these, say by electron microscopy, you see that the surface is very ragged. It looks like what we can expect from a polymer brush. And the most interesting thing is that these surface-grafted polymer beads sort of act like bacterial flypaper. If we just mix a small amount of bacteria with these beads, and these are again 5 micron beads. We've done some work with the 300 nanometer cationic beads, but what you can see is there are a number of bacteria associated with the bead, even at fairly low levels of the beads and fairly low concentrations of bacteria. We've seen some of this on flat surfaces, but it's really dramatic in these cases. And so what we've done is to actually study the bacteria with these 5 micron beads and look at the biocidal activity in some detail. And we've both cultured the bacteria, but we've also used what are called live dead assays, where we treat the bacteria with a mixture of 2 dyes. There's a red fluorescing dye, which can get into either live or dead bacteria and bind with the DNA. And there's a green fluorescing dye that we're using that enters only when the membrane is permeable. In other words, the bacteria are non-viable. And the results are really quite nice. Here are different confocal fluorescence images. Here we look at the polymer fluorescence. Here's a wavelength where the red dye emits. Here's the wavelength where the green dye emits, and then this is a composite. And so you can see clearly the polymer bead, which is surrounded by several bacteria. Typically we find 7 or 8 will surround the 5 micron bead. And you can see this is where the bacteria and the beads are incubated in the dark for an hour. And you can see there's no evidence of killing of the bacteria. Viable bacteria are seen here, and also viable bacteria that are on the beads. So just attaching the bacteria to the beads doesn't kill them. On the other hand, if we irradiate them, this is with visible light, for about 30 minutes, what we find is you can see the green, the signal for dead bacteria, as well as the red, so you get both red and green, and you can see the composite. And I don't believe any of these bacteria on the bead are viable after a short amount of radiation. So we think that these surface-grafted conjugated polymers can be very useful for biocidal activity. To just summarize here, bacteria plus visible light, no killing. Bacteria in the dark, no killing. Bacteria in the beads in the dark, no killing, at least on a short term. But visible light, bacteria and beads, and the bacteria are killed. They may be even degraded from what we've seen with some flat surfaces of this. So we think these polymers grafted can be very useful. They're more stable than coatings. Synthesis is straightforward. We think we can tune both the dark and light-induced activity, and we think perhaps that this will be very nice to give us liquid-like behavior in a solid sample. We'd like to be able to control the balance between entrap and release. In other words, we don't want to build up biofilms that render these beads or surfaces not useful. We're really interested, finally, in reactivity towards other agents and the possibility of using these to sense and destroy bacteria and other pathogens. Thanks.
Questions? Paul?
What do you consider other silica objects? You know, silica tubes are now available, cylinders, and whether these might, you know, have also additional preference for certain cells.
Yeah, we'd like to put them. We'd like to build planar surfaces, planar silica. But yeah, I mean, we could use a variety of things. We're interested in filtration. We're interested in the possibility of putting these things into something like a paint. But we're interested in coatings that both would be wet and dry.
So there are, you know, a lot of these paint techniques that use mica sheets.
Yep.
And this company Merck, EMD Merck in Europe, is an expert at putting, in addition to mica, putting other things on top of it.
Mm-hmm.
And maybe they have technology to do silicon.
I would suspect there are. Andy?
Dave, on the assays you showed with the lipase activity, when you form a membrane on these polymer surfaces, is it clear that it's a single bilayer that goes down, or is there some possibility of a multi-layer membrane?
It could be a multi-layer. We don't know. We essentially make vesicles and then sonicate the beads in the presence of the vesicles. And you know, we can't rule out there may be patches. I wouldn't say we can guarantee that there's a simple bilayer. Yeah, Victor?
I still don't quite understand how this super quenching works. So I'm trying to connect Paul's story and your story.
Okay.
So Paul is saying, so there is a low energy site on the polymer, excitation eventually finds it. How metabiology finds this excitation site?
Oh.
Metabiology, you're quenching it.
Oh, yeah.
How does it find it eventually? Because it's charge, eventually what quenches is charge transfer. You break the exciton, right?
Well, we've done a lot of studies to try to unravel the mechanism. For example, we've done work with cyanine polylysine. So we've made cyanine polylysine isn't a conjugated polymer, but it gives very similar behavior in terms of super quenching to the conjugated polymers. And there, what we find is we can start with an oligomer. We start with 1, 5, 12, and so forth, and we can put make it larger up to I guess 1,000 or more repeat units, and we can just measure the quenching that occurs, and then we see that for example, we get a quenching enhancement. We see where it levels off, and it's about about for the cyanine polymer, it's about 100 repeat units per quencher. If we look at the repeat units, so one quencher is somewhere, it has to be within 100 units to get quenching. I think it's very similar.
But some of the dyes which look like charged, which did accepted energy but didn't emit, would have absorbances that would overlap with the emission from your polymer.
Yeah, we've tried.
Some of their dyes, I mean, I think probably knows it, but some of the dyes
right? So that's
are good
essentially
acceptors. Is it possible that there's any sequential buildup, of the system? In other words, where some chemistry happens and then the system itself modifies to quench even more?
We haven't seen real evidence of that.
This is all instantaneous.
Yeah, and it seems like the level of quenching remains constant in time.
If
There are no additional questions. Let's thank the speaker again.